rule, which transmits the genetic information from DNA to protein. However, is there any other rule that RNA can play on in the cells, or what can RNA do for us? So it also plays roles in regulating gene expression. For those RNA that's not for protein coding, we call the non-coding RNA. The regulatory roles of RNA has been described in the past 3 decades. In 1980s, the discovery of a ribozyme by Tom Cech's group describe the function of ribosome. It can function as an enzyme. It itself, not the protein, can function as an enzyme. And also in 1990s, the discovery of RNAi phenomena also give us a very interesting example. Example of regulatory role of non-coding RNA in the cells. So today we know that for those RNA not for protein coding, is the non-coding RNA. What's the function of those non-coding RNA in gene expression? So in cell, we can classify this non-coding RNA based on the size. For the long non-coding RNAs, we call link RNA, is the non-coding RNA over 200 nucleotide in length. For example, in the nucleus, 7SK is a non-coding RNA which associated with transcription factor and regulate the transcription by PO2, RNA polymerase II. Another good example is for mammalian X chromosome inactivation. 6 is the long non-coding RNA expressed from the X chromosome, and it involved in X chromosome inactivation. For those non-coding RNA in short length, we call small non-coding RNAs like microRNA and siRNA. It functions in post-transcriptional gene silencing, or we call the PTGS. So today our lecture will focus on the small non-coding RNAs in gene silencing and post-transcriptional regulatory mechanism. So far, there are many different types of small silencing RNA has been discovered. This review shows that such as a microRNA, or here endo or exo siRNA, or piRNA. However, other than those, also have the CASI or TASI or NATSI. That's all from different species, and also they have other features in with different length or associated with different kind of proteins. In human, we know that those small RNA regulate the gene expression at the various steps. In the chromatin modification step, some of the small or large non-coding RNA can promote the formation of the heterochromatin, like the 6th in X chromosome inactivation, or in piRNA, or blocking the transcription. For microRNA or siRNA, it functions majorly in the cytoplasm and block the translation of a specific mRNA. Also, for those microRNA, siRNA, if it's perfectly matched to the mRNA sequence, it will trigger the specific cleavage and follow up by mRNA degradation. So will trigger the mRNA decay. In today's talk, we will talk about the small non-coding RNA in human cell. There are 3 classes of 3 small RNAs in human cell. Today we'll focus on those 3, but due to the, I think, the limitation of the lecture, because this year they kind of we shrink the lecture hours. So maybe it depends on how much I talk in the lecture. I might skip the piRNA part. Okay, and so I focus on the siRNA and the microRNA mechanism. So we know that the siRNA is the single-stranded RNA with a length of 90 to 21 nucleotides. It functions in target the mRNA cleavage. And for the microRNA, it's endogenous small small RNAs and with a length of 19 to 25 nucleotides. It involved in translational repression and also trigger the mRNA decay. For the PV interacting RNA, we call piRNA, is a little bit longer, and majority is in with a length of the 24 to 31 nucleotides, and involved in transposon control and some of the transcriptional silencing mechanism. And now some of the piRNA only found in the germ cell and also for some cancer stem cells. In the RNAi pathway, the long double-stranded RNA or short hairpin RNA here can be processed by dicer. So dicer is the endonuclease, double-stranded RNA endonuclease. It will cut the double-stranded RNA in the exact size, like this dicer tool, like you sometimes use in the kitchen. So it will cut those RNA in the exactly the same size. So we call it dicer processing, and then make the double-stranded RNA the 21 nucleotide duplex with 5' phosphate and 3' 2 nucleotide overhead at the 3' end. 2 nucleotide overhead at the 3' end. This is the RNAi pathway. So for the endogenous expressed or overexpressed from the construct or short helping construct, it could be processed by dicer and followed by risk assembly. Similar to this mechanism, we can also chemically synthesize the siRNA duplex, and the transfat or introduced into the cells will undergo the 5' phosphorylation and the risk assembly, similar to the endogenous double-stranded RNA processing, risk loading processing. So what is risk? Risk is RNA-induced silencing complex. In the RNA-induced silencing complex, first the double-stranded RNA has to load onto it, and only one strand will stay on the risk. So this strand we call guide strand, which guide the risk to the targeted mRNA or target sequence, target the molecule. So this step is called risk formation or risk activation. Only the risk associated with single strand is the active form and will recognize the target mRNA. For the perfect match, the risk in the siRNA pathway or RNAi pathway, it will trigger the mRNA cleavage and then followed by some exonuclease in the cytoplasm. So the mRNA will be degraded. Then the active risk can recycle for next round of target recognition, then amplify this continue the cleavage activity. For the microRNA pathway in mammalian cells, the microRNA genes were transcribed by RNA polymers 2, and then form the primary microRNA or microRNA precursor. Those microRNA precursors include a very long stem and a lobe structure are processed by Joshua DGCR8 complex in the nucleus. Then and form the helping structure. Those helping structure can be exported by exporting fine GDPS to the cytoplasm. So in the cytoplasm, it will continue for the microRNA biogenesis, similar to the siRNA, shRNA or long double-stranded RNA. It can be processed by dicer to generate a double-stranded intermediate and followed by RISC assembly step. For the microRNA RISC, we call MI RISC, is the risk associated with microRNA. It's similar to siRNA RISC. It will recognize the target. However, because the a mismatch at the central cleavage region or some other region is not a perfect match to the target mRNA, it will not trigger the cleavage. Instead of triggering the mRNA cleavage, it will repress the translation. So this is a microRNA pathway. So if we compare these two pathways side by side, we can see some similarity or they share the same feature. First, they all processing by dicer, meaning that for the activation of the siRNA or microRNA, those small RNA mechanism, it has to be a short duplex at the beginning. And then the next is it included the RISC assembling and the function step. So on the double-stranded RNA of the double-strand product by dicer will be processing by the RISC and on one follow the unwinding step. Only one strand stayed on the organelle protein in the RISC. So let's zoom in the biogenesis of microRNA. Like I just mentioned that the microRNA was processing, the microRNA was transcribed mostly by RNA polymers too, which means like most of the protein coding mRNA, it is capped, fibron capped, and with the poly A tail. And it will express as a cluster, meaning that one transcript includes multiple microRNA for the following processing, and it will localize in the intergenic or intragenic of the genome. Or it can originate from some protein coding, the intron or exon of protein coding gene or non-coding RNAs. So here are the genomic locations and the gene structure of human microRNAs. For those, some of the microRNA can localize at the non-coding transcription unit with intronic microRNAs. Like this one, the DLU2 is a non-coding transcription unit. This is a splicing feature. Then mere 15A and 16.1. -1 is localized the intron of the non-coding transcription unit. But it could be localized at the exon of the non-coding transcription unit, like the big here, the mere 155 is the exon region of the non-coding transcription unit. It also can localize at the intron of the protein coding transcription unit, like MCM7 gene. The intron, one of the intron contain the cluster of the mere 25, 93, and 106B microRNA, and also can be at the coding region. It's very some of the microRNA can be found from the coding region of the transcription unit. So those are the genomic location of the gene structure of microRNA. Then how to generate those microRNA in the nucleus? Like I mentioned, in the nucleus, the nuclear processing, including the cutting the cluster to become a long helping microRNA, pre-microRNA. So it's a primary microRNA to pre-microRNA step. It was processed by DROSHA and the DGCRA complex. In this complex, we call the microprocessor complex. Is the protein complex that can recognize the specific structure of the hairpin and measure exactly the precise site for the cleavage. However, some of that not follow the microprocessor mechanism. One group of a microRNA called neutron is the pre-microRNA direct splice out from intron without the microprocessor. Okay. Or for those after the nuclear processing or before the nuclear processing, it could be edited by the RNA editing mechanism through the either A to AI RNA editing. So here are the structure of those enzymes involved in microRNA biogenesis. The first is human drosha. We can see it includes two RNS3 domain here, and also a double-strand RNA binding domain. For human, DGCR8 is another protein associated with drosha, but we can see it contains only the double-strand RNA binding domain, no RNS3 domain, which means that this protein is facilitating the drosha function, but the enzymatic activity is located at the drosha. So here showing the microprocessor. The DGCR8 complex with the drosha, and the DGCR8 recognize the branch site of the basal of the stem loop structure. So it recognizes the branch site here, the basal branch site of the stem loop structure. And because the drosha interact with DGCR8, it will exactly cut the double-stranded RNA from at the 11 base pair from the basal branch site. So it looks like they serve as a molecular ruler. So meaning the rate measure the distance of cleavage for drosha to make the microRNA precursor, to make the pre-microRNA. And the third one is the human dicer. We know that it's involved in the double-stranded, long double-stranded RNA and the helping RNA processing to cut into 21 nucleotide duplex. Similar to the drosha and the DGCR8 here, it contains RNS3 domain here, RNS3A and RNS3B. So sorry, it's not clear. The green dot here showing the catalytic site, and there are two catalytic sites, and it's kind of a slice out. So that's why the product of the dicer is making the 3' the two nucleotide overhang at the 3' end, because it's not parallel to each other. So it's they contain the slight two nucleotide overhang. And also here the drosha has a symmetry cleavage, so that's making the another 3' overhang here. And the pass domain is to recognize the free 5' end phosphate. Therefore, because the distance of the drosha structure from the pass domain to the RNS3 domain, it will generate the double-strand RNA in the almost exactly the same size. So review those biogenesis of microRNA transcribed by RNA polymers 2, majority of by PO2. Some of case is from by other RNA polymerase, and then processed by drosha and the DGCR8. In fly, DGCR8 also called pasha, meaning the partner of drosha. Okay, then crop the to generate the pre-microRNA exported to cytoplasm, and followed by the dicer processing. Okay, in a fly, it's dicer for microRNA. However, in human, only one dicer for the microRNA siRNA processing. And it also associated with some argonaute protein. Those argonaute protein is the risk, the key factor in the risk, meaning that after the dicer cleavage will directly followed by the risk loading risk loading step. Some of the protein also associated with the dicer, like the TRBP and the PAX. And those interactions will help the transfer of the double-stranded RNA from dicer to the argonauts protein, and then followed by the risk loading step to generate a mature microRNA on argonaut protein. So those canonical microRNA pathway can also happen simultaneously with the splicing phenomena, like the canonical intronic microRNA. It was processed by the Joshua complex and cropped to generate the pre-microRNA. And at the same time, the spliceosome complex also can trigger the splicing, so they can generate a mature mRNA parallelly. However, in some non-canonical intronic small RNA, those small RNA sequence is very close to the branch site or some of the spike branch site or lariat in the lariat of the splicing product. So that the mechanism is after the splicing, those branch, the pre-neutral, will access the branching to generate, reform the pre-microRNA without the DGCRA Joshua complex cleavage, and or will trained by exonuclease to generate the pre-microRNA helping structure and followed by the mature microRNA biogenesis. So this non-canonical intronic small RNA we call neutral is the biogenesis of microRNA bypass microprocessor. So then after generate those pre-microRNA and delivered, exported to the cytoplasm, what is the next step? In the cytoplasm processing, it was cleaved by dicer to generate the 21 to 24 nucleotide duplex from about 17 nucleotide primary pre-microRNA. And after dicer cleavage, the next is risk loading to remove the passenger strand microRNA. Actually, we can call that, we just name the passenger strand because the other strand is stay on the risk. For those strands to be removed out, we call passenger strand. But in the biogenesis of a microRNA, we can see that it's duplex. So only one strand can stay on the risk, the other should be removed. But which one should stay and which one should be removed? There are a couple different mechanisms. There's no exactly which strand is always be the one. So if you sequence the microRNA in the cell, sometimes you can see both strands stayed on the risk. And only the mature microRNA stayed on argonaut protein. So here show the dicer processing. Like I mentioned, the dicer also contain the RNase3 domain A and B here, and it will cut the double-strand RNA to generate a 3' overhang. And here is the pass domain, and because the distance from pass to the catalytic residues, the product of dicer processing is always 21 nucleotide in length. So then we move on to the siRNA and the microRNA effectors, which we call the risk RNA-induced silencing complex. People were curious about what's the exactly composition of the risk. So about 10 or 15 years ago, when the people discovered the siRNA and the microRNA mechanism, a lot of lab work on the biochemical isolation of a risk. And they found that even from the fractionation and all other purification mechanism, it's kind of a huge variety of different RNPs. So sometimes it could be from the modest size, about 150 kilodalton, and even to a huge about the ATS, which means 3 megadalton particle, and also contain the risk, meaning that it could also have other intermediate size. So it implied that in cell, maybe in different situation or different compartment, the risk interacting protein and the complex are different, and it could vary the exchange their interacting partner for specific function. However, based on the small RNA associated on the risk, we just simplify the risk as the SI risk or micro MI risk based on the small RNA that associated on it. The key factor on the risk is the argonaute protein. Like in the previous slide, I show you the AGO1 or AGO2. Okay. The argonaute protein is an exact slicer of the SI risk. So in human is AGO1, which has a catalytic activity. In human is AGO2, sorry. AGO2 is a catalytic argonaute catalytic risk key factor of the risk. This is the structure of the AGO2, and it contains a pass domain, mid domain, PV, and the N-terminal domain. Also showing that this is the target mRNA, and the yellow line is the siRNA associated on the argonaute protein. So pass recognize the 3' end of the siRNA look like a hook the strand here, and the shape is more like a croissant, and in the central pocket, it has the space for mRNA and siRNA recognition. So, and the PV domain is the catalytic domain, contain the endonuclease activity here. So from this side, look at the structure. So from if you look from the left side, look into the central, looks like a hole and the two like a croissant and a clip on the double-stranded structure for the siRNA and the mRNA target. And here is a catalytic site, the PV domain, and the 5' end of the guide. So, which means that it has to be a very perfectly matched from the 5' end to the cleavage site. And this indeed is the seed region. We will talk about it later for the microRNA and the siRNA recognition, because the only perfect match at this region is sufficient to provide the specificity of the target recognition by the risk. So argonaute protein, how many argonaute protein in nature? Actually, in human, there are 8 argonaute proteins, 4 argonaute and 4 PV protein. So difference in different organisms. In yeast, only 1 argonaute, but however, in worm, the C. elegans, there are 26 argonautes. And those argonaute protein can be classified in 3 classes, including one is argonaute family, the second is PV. PV, this term comes from P element, induce wimpy testis of phenotype of fly with the PV mutant. The third one is a worm argonaute, we call Wagos, because the C. elegans has very unique argonaute protein that was not there's no homologous proteins in other species. So this is just a very simple phylogenetic tree of argonaute protein. Here showing the black is the Argonaut family, and the green is the peewee. And all these red lines show the Wagos. So, meaning that those Argonaut has high similarity and peewee, and it can be classified in 3 clades. So, like I say, in mammalian cells, we have in human we have AGO 1, 2, 3, 4, and the peewee protein we have HeWe1, HeWe2, HeWe3, and Heli. So for AGO 1, 2, 3, 4, only 2 contain the catalytic activity, 1 and 3 and 4. It can associate with microRNA. However, the expression level is relatively low, and it doesn't induce the RNAi activity, cannot cut the target if it is perfectly matched. Also, because we want to know the risk component, the people identify those proteins associated with AGOs, such as in AGO1, people found dicer on the DCP and MOF10, those protein interact with AGO1. For AGO2, a lot of other protein interact, like GW182 or TNRC6A. I mentioned that because later on we will talk about those proteins in the microRNA mechanism, because it's kind of bridging the microRNA to decapping the adenylase complex. And also DCP1 and A for decapping, because microRNA and the AGO localized at the cytoplasmic P-body for the RNA decay. Some of the protein like RCK-PP4, we know that DDX6 also interact with the DCP and the AGO2 and other proteins. So back to the risk and the risk loading activation. So far there are 2 mechanisms for the risk loading. The first is for the perfect match, the duplex. During the risk loading, Argonaut will cut the passenger strands and then release it out. And the other is for some of the microRNA plus precursor, it contains the bulge or mismatch at the duplex of the microRNA, pre-microRNA. Then during the risk loading, the AGO cannot cut the passenger strand. Instead of cleavage, it triggers the unwinding and release out the passenger strand. And interesting that the risk loading has some kind of strand preference. It was determined by the stability of one strand, one end. If the one end is contain the more AU base pair, it's more easy to unwind. There are other proteins to help to determine the orientation of a risk loading for those double-stranded RNA. And after the risk loading, the single strand on the single strand microRNA on the risk complex will recognize the mRNA, target mRNA. During the target recognition, we can zoom in and hypothesize as a multiple step. First is the C region, like I mentioned in the previous structure slides. In the C region, the perfect match will help the duplex form and twist the duplex formation, and to provide beta recognition of the duplex from the Argonaut protein to its target mRNA. So if it's a perfect match and its base paired at the central region, will trigger the cleavage by AGO2. So this region is very important, the C region of microRNA, and most of the time we will see those microRNA recognize the 3' UTR of the target mRNA. David Batel's lab provided some algorithm to predict the microRNA target. They published this and generate the powerful tool called Target Scan, and it's available from their website. So from their prediction and their experiment, they clarify that those microRNA target site are a canonical site, including C region at the position 2 to 7 from the 5' end of microRNA. Those are 7-mer A1 site, meaning that the first site has to be A, and other are 7 plus M8. So those seed region all contains the perfect match from the microRNA to the target. And sometimes if the seed region is shorter, in some cases also can be recognized by the microRNA. For the atypical site is, in addition to the seed region, at the 3' end of microRNA also have several base pair to the targeted mRNA. Those are 3' complementary sites for the atypical site of the microRNA recognition. So here shows that multiple many different kinds of microRNA target prediction tool from different labs. Some of that including the different species database from mouse, fly, or worm. And right now they have expanded their database as well. So if also all those databases has been integrated in the Nearbase website, supported by Sanger Institute. It provides a lot of information of current identified microRNA, including the from different species and all different variant form. Then we go back to the function of microRNA. What's the functional mechanism of microRNA? We know that how they log onto the risk, how they recognize target, and then how do they trigger the gene silencing mechanism? To understanding the mechanism of microRNA, people try to zoom in at what kind of protein interact with microRNA or argonauts. So identify the interacting protein like the GW182 or RCK or called DDX6 or DCP12 or EIF6. And the other way is to measure or monitor where those microRNA localize and where the argonaut protein localize, the risk localize. It's interesting that the AGO2 localize at some specific foci in the cytoplasm. Those foci are P-body, the RNA processing body. But if we enhance this is the like a low intensity detection. If we increase the gain of those fluorescence signal, we can see it kind of spread out cytoplasm, and even in some cell, we can detect the AGO2 in the nucleus. And also some biochemical approaches prove that those argonaut protein can also be identified in the nucleus. And so although we know the major mechanism of the microRNA is to block the translation, so it should be happened in the cytoplasm, but we cannot rule out that it can also translocate in the nucleus, or even for those circulating RNA story, the circulating microRNA. We know that microRNA can be released out to the blood and as the small exosome. Okay, so exosome can contain the AGO2 protein and also the microRNA, and so can be extracellular microRNA and release out into the blood. And I'm sorry, could be probably not clear enough. So here showing the AGO2 signal here, and then also AGO3 and AGO4 all localized at the P-body. So now the current model of microRNA-mediated gene silencing in animal contain many molecules, many proteins. If we start with the AGO and the microRNA target recognition at the 3' end of the mRNA, the AGO interact with GW182 and the GW182 recruit the poly A binding protein complex complex C1, PABPC1. Then this trigger the deadenylation by CCR4 and CNOT complex. In addition, those also interact with some decapping complex, then reach to the 5' end of the cap site and trigger the decapping, followed by the some exonuclease from the 5' end. So this is the current model of microRNA mediated gene silencing. In addition to deadenylation and decapping, also could repress the translation by multiple mechanisms. So here is a zoom in of the complex AGO and the microRNA here, and the GW182 or called TNRC6 protein here interact with PABP and bringing the CNOT complex, then EDC3 and DX6 and hook on the this decapping complex. It kind of linking all the RNA quality control or RNA decay complex, and those DCP and DDX6 all localized, and the AGO to all localized at the P body for the RNA processing, RNA storage and the decay. But in addition to those, other lab also suggests some mechanism. So there's no exactly one mechanism. Those can happen simultaneously. So AGO2 can block the entrance of the 40S or 60S for the initiation of translation. Or in some lab, they show the interaction with the microRNA and the risk at the 3' will trigger the ribosome early drop-off. So meaning that the translation efficiency is decreased if the microRNA recognized to the 3' end of target mRNA. Another mechanism is linking the decapping complexes. So once the microRNA interact with the target mRNA, the decapping complex bring in and trigger the 5' to 3' decay. And those can store at the P body for mRNA storage or mRNA degradation. So I think I'm probably going to skip the PV protein and the piRNA part. Just quickly showing those piRNA and the PV protein is another clade of Argonaut protein. And in mouse has been identified many in the early the embryogenesis or spermatogenesis or genesis, there are a couple PV protein involved in the germ cell formation. Okay. So sorry, I have to skip those parts. Maybe I will leave some time for the question, just Q&A. Then. So this is probably what I have today, because we have the paper presentation. So I will skip the piRNA part. Summarize today's talk here. We know that the siRNA and microRNA pathway is the small regulatory pathway in animal. And today I talk about the microRNA biogenesis, and I introduced some key proteins involved in siRNA and the microRNA pathway, including dicer, DGCR8, dicer, DRASA, Argonaute proteins. And I introduced some mechanism of the microRNA median gene silencing. And sorry, probably don't have enough time for the piRNA in germ cells. So thank you for your attention, and I'd like to take a question for today's lecture. Any question?
Any questions from Kyoto or Tsubasa?
Actually, I have a question from the Tsukuba side, and I have a question about the microRNA function. So the page 31 the PDF file.
What? You know, microRNA function in what?
MicroRNA function in the gene repression.
So,
so the next one.
This one? Or this one?
No, next.
This one?
Sure. Yeah. Actually, so the post-initiation inhibition. So I'm wondering how so the miRNA complex inhibit the translation.
Yeah. Okay. So, I have to say there are very little evidence for this, but because this paper was published by a very famous RNA group, so they proposed that maybe interacting with the microRNA can induce some factor that block the ribosome or let it slow down during the translation, or even some of the microRNA can recognize the protein coding region, because we cannot really rule out that microRNA not recognized to the protein coding region. So it could be another mechanism, but this is not the major theory or concept aspect for people to study the microRNA mechanism, but still have some evidence to support this. Okay. So I don't really know how this happened, but right now the main focus is on this package and the GW182. Is it okay? Sorry.
Yeah. Okay. Thank you very much.
You're welcome. Any other question? Taiwan side? No? So it's what? Where?
This is Kyoto side.
Yes, please.
Yeah, I have a question. So,
sure.
Because you said there are several Argonaute homologs in fly or other animals.
Yes.
And you said mainly angle 2 is active.
Yeah.
And other angles have weaker function or something.
Yeah.
And but still, I know in fly, angle one.
It's for microRNA. Yeah.
Yeah, angle 1. Angle 1
is a mutation.
Showed some phenotype. So,
okay. I'm sorry, I didn't mention clearly. In mammals, AGO2 is the major one, but in fly, the AGO1 and the AGO2 is parallelly. AGO1 is specific for microRNA, and the AGO2 is for siRNA. So in fly, the microRNA mutant, the AGO1 mutant will represent the misfunction of microRNA. So in fly is AGO1 for microRNA and AGO2 for siRNA, but in human is only AGO2 is the major one, but still have some microRNA associated with AGO1, 3, 4, but this is not the main focus for people to understand the mechanism. So sorry, I didn't mention clearly in the previous slide. Yeah.
Okay, thanks.
Okay, thank you. Any other questions? No.
Another question from Kyoto side.
Yes, please.
I was just curious if there's any difference between proteins that are going to be secreted versus proteins that are intracellular with regard to silencing mechanisms. Is there any difference at all between those RNAs?
Oh, yeah, it's a good question. So people are still working on the exosome RNA interacting protein and all those different proteins in the cytoplasm. But at least in the exosome, I think the Dr. Shen is here. Then he is the expert for the exosome. AGO2 was identified in the exosome, but maybe for the secretory microRNA, it has to recognize some intracellular, some endogenous or cellular mRNA. So if it recognize another cell and communicate with other cell, it will fuse to the target cell. Then the target cell also have their own RNA, microRNA protein, machinery protein. So maybe trigger another similar mechanism, but maybe it's not really function in the exosome, but is during the circulating may recognize other cell for its function. But this is just my hypothesis. I don't know if there are any detailed mechanism happening in the exosome. So it's okay. Hi, James.
Can I ask one more question?
Sure.
I think there's kind of this hot topic of transgenerational epigenetic inheritance, and some people have speculated that it might involve miRNAs. Do you have any clue about which mRNAs might be involved in this?
Excuse me, sorry, because the sound is not clear. Could you repeat it again?
Yeah, some people have speculated that the phenomenon of transgenerational epigenetic inheritance might involve miRNAs. Do you want to speculate a little bit about which ones might be involved?
I don't know. Yeah, because I know in worm, in C. elegans, the siRNA, the small RNA can be inherited to their offspring. The RNA can be inherited to the offspring. But so far we know that in the germ cell formation, we know some of the, because in the, let's say like the spermatogenesis, those RNA and RNP, they will form the very huge chromatoid body. Those chromatoid body like the P body, but it is kind of all like wrap up the RNP and also including argonaute and the dicer. So of course, some of the microRNA could be packaged in the P body or the chromatoid body, and maybe in the spermatogenesis, the sperm may not carry the chromatoid body, but in oocyte, the egg will contain the chromatoid body, so may have some microRNA packaged in the oocyte. So it will be very interesting to like collect the oocyte and just analyze the microRNA. But I have to mention that the argonaute, the microRNA and siRNA mechanism are kind of silenced or repressed in the early development. So almost no siRNA can function in the embryo. That's why when some of lab using the zebrafish and try to do the knockdown, but never for the early stage of embryonic development, it's not working very well. So maybe the microRNA can transmit from the oocyte, but the onset of the function could be later. But I don't know if there any evidence showing what kind of microRNA has been identified. Okay.
Good explanation. I think we agree.
Thank you.
The mechanism is still very, very mysterious.
Yeah, that's true. Thank you. Any question? No question from Taiwan side? If no, let's take a 5 minutes break and for students to set up the presentation. Thank you. Thank you. Okay. Let's come back to today's journal club, the paper discussion. Today we have 5 students from NTU side. They are going to present an article published in Cell Report and talking about the RNAi factor presented in the nuclei, human cell nuclei. Let's welcome them.
Yes, thanks for Professor Zhu's introduction. And today we're going to talk about RNAi factors are present and active in human cell nuclei. And during the cytoplasmic RNAi interference, and there are 2 well-known pathways, before the course talk about the microRNA pathway and siRNA pathway. And these small double-strand RNA will cooperate with the RNA-induced silencing complex. We call it RISC. And then the RISC will target the mRNA for the post-transcriptional regulation. And in the nucleus, the primary microRNA are transcribed by RNA polymerase II. And with the Droser microprocessor, the primary microRNA going to the precursor microRNA, and then with the exporting 5, and this protein will transfer the precursor microRNA into the cytoplasm. And in cytoplasm, the dicer will cut the precursor microRNA into the microRNA duplex, and with AGO2 to form the precursor risk. Then one of the strand is the passenger strand removed. Then the precursor risk become mature. Then the guide strand will lead the risk complex to the mRNA and for the degradation. And siRNA pathway is that the double-strand RNA are processed by dicer and into a double-strand siRNA, and then with the AGO protein become the precursor risk. Then finally, the function is that one of the strand is cut by the AGO protein, and then the SI risk will target the mRNA target for the mRNA cleavage. And there are several factors have been implicated the loading of the risk and the risk activation. The first one is HSP90 and HSC70, and some evidence shows that these chaperone-like proteins will open the AGO protein to guide the initial binding of the duplex microRNA. And there are also co-chaperones like CDC37HA1, FKBP4 and 5, and P23. And another one is a C3PO complex, which consists of tracylin and the TRAX. And these two become a C3PO complex. And in RNA interference, this complex will help the unwinding of the siRNA duplex and remove the passenger strand. Then the precursor risk will become mature. And all of these RNAi interference and its silencing activity is occurred in the cytoplasm. But RNAi can function in the mammalian nucleus and regulate process like transcription or splicing has remained unclear. And some laboratories have suggested that AGO2 and other RNAi factors can be found in the nucleus, like dicer1, and it's associated with the nuclear pore complex protein, NUP153. And dicer is also a shuttling protein, means that can transfer between the nucleus and the cytoplasm. And RISC can also shuttle in between the cytoplasm and nucleus. And AGO2 is not only localized in the cytoplasmic, but it's also to the nucleus. And importin 8 is, and it's required for the microRNA guide gene silencing and affect the nuclear location of AGO proteins. And there is some evidence shows that siRNA have been reported to silence the nuclear enriched small nuclear RNAs, 7SK and U6. And microRNA have been also found in the nucleus, but their biological roles are unknown. And the study have shown that maybe the RNAi is in the nucleus, but there are also some controversial point because we don't know the purity of the cell extract or the resolution of the localization study is not well, and nucleocytoplasmic transport of the RNA target and product of RNAi are also a controversial point. So the study today is want to investigate the localization, interaction, and the activity of the non-RNA factors in human cell nuclei. So they use some tools. They use HeLa cells to find the localization of the AGO2, and they use the technique called immunoepifluorescence microscope. And this technique is help them to rapid and find a 3D image of the nuclei. And then they also use confocal immunofluorescence microscope for comparison. And second, they use decided protocols to facilitate entry of the antibody into the nucleus. And this is the result that showed in the nucleus, which is the blue one is DAPI, means in the nuclei. And you can see the green one, AGO2 protein, is localized in the nuclei. So in this 3D image, you can see the RNA factors AGO2 is within the nuclear compartment. And this video also shows that the AGO2 is in the nucleus. This is a 3D imaging video.
So next, they want to further confirm the nuclear localization of AGO2 or other RNAi factors. So they use cellular fractionation and Western blot to check the AGO2 and other RNAi factors actually expressed in the nuclei. So he developed a stepwise protocol. He can separately isolate cytoplasmic, whole nuclear, and nuclear plasmic, and chromatin associated fraction. And this fraction will use for various assays, including protein and RNA detection, chromatographic or biochemical fractionation, and enzymatic assay. But as RNAi factors can also localize to ER, it makes detection of RNA factors location in nuclei tend to be difficult because the size of ER membrane is too close to the outer membrane of nuclear. So they try to use different detergent and condition to prepare nuclear extract. First, they wash nuclei with buffer contain different detergent, including Twin 20, Triton, and NP40, and then check the nuclei and ER by fluorescence microscopy. We can see the best efficiency of removing ER protein with 0.3% NP40, and here show almost no yellow signal of ER tracer. And we can see this Western blot data. The correticulin is ER lumen marker and chromatin is ER membrane markers. And the data show that there is no ER lumen and ER membrane protein contamination in nuclear extract. And next, they move on to check for expression of RNA factors in different cell lines. The Western blot shows that in different cell lines, RNA factors expression including AGO2, dicer TRBP, and TNRC6A, also shown in the nuclei. And we can see in nuclear extract of these 4 different cell lines, there are no cytosolic marker tubulin and ER marker, carotene and mitochondrial markers as force in a combination in this nuclear extract. So this data show that they got cytosolic extract and nuclear extract with no contamination. And quantitation of this Western blot reveal relative nuclear abundance ranging from 50% to 40% to 50% of these RNA factors. In the past, it's difficult to clearly observe the localization of RNA expression because of the incomprehension of cytosolic and nuclear fractionation. So they set up this protocol to certainly separate the cytosolic and nuclear fraction. So according to figure 1 and figure 2 data, it suggests that these RNA factors are indeed present in nuclei extract.
After knowing that these RNA factors are present in the nuclear extract, they also want to know whether these RNA factors in the nucleus interact with each other. So they use the method to test protein and protein interaction. It's called immunoprecipitation, called IP. First, we got a nuclear extract from the nuclei and with our interest protein inside. And then we put the first antibody to do immunoprecipitation to pull down the protein, and then use the second antibodies to do Western blot analysis, so we can know whether these proteins interact with each other. This is the data they do. Okay. First, they can know that the ARGO2 can interact with dicer and the TLR6A. And dicer can also interact with ARGO2 and the TLR6A. And then TLR6A is also interact with ARGO2 and dicer. And next one is TRBP can interact with this protein. So we can know that these RNA factors in the nucleus can interact with each other. Later, they want to configure that again. So they generate a T47D cells stability express flag HA tag ARGO2. And then they show that the HEA cell is a negative control. The data shows that these RNA factors are interacting with each other. What's more, they want to know whether this protein interaction is dependent of RNA or not. So they treat RNAs to do co-IP. This result shows that the association of RNA factor is independent of RNA. To visualize the association of TLR6A and ARGO2 inside the cell nuclei, they perform immunofluorescence microscopy. This blue one is DAPI, means the nuclei, and the ARGO2 is a green one, and the TLR6A is a red one. We can see this protein overlap in the same location. So they saw that this RNA factor in the nuclei can perform complex. After that, they also want to know if there are differences between the nuclear factors complex in the nucleus or in the cytoplasm. So they do size-exclusive chromatography and ion exchange chromatography. This result shows that the RNA factors in the nucleus and in the cytoplasm are very similar, except the TRBP. TRBP will elute later in the cytoplasm. Maybe it has a smaller molecular mass. What's more, they also do the ammonium sulfide precipitation to test the stability of this complex. This result shows that the RNA factors in the nucleus and in the cytoplasmic separate use a different concentration of ammonium sulfate to separate this protein complex, so they may have different characteristics. All these results demonstrate that nuclear RNA factors can form a multi-protein RISC-like complex, and the characteristics are a little bit different from that in the cytoplasm.
So after seeing that the RNA factors are present in the nuclei, the author want to know that whether they can also silence the nuclear RNA. So first, they quantify the long non-coding RNA that are primarily associated with the chromatin. Here, it is represented by the MALAT1 and NEAT1. Just mention that the MALAT1, also known as NEAT2, the NEAT stands for nuclear enriched abundant transcript. Just as their name has implied, they are highly expressed in the nucleus. And for comparison, the mRNA that was primarily present in the cytoplasm are represented by LPR13 and PPIA. So after the siRNA treatment, the MALAT1 and NEAT1, which primarily present in the associated with the chromatin, was reduced after the siRNA treatment. And the mRNA, RPL13, and PPIA, which primarily present in the cytoplasm, are also reduced after siRNA treatment. So this result indicates that the siRNA functions are similar in cytoplasm and nucleus. And then to further test whether RNA silencing in nucleus is due to AGO2 cleavage, mediated cleavage, they perform a 5' race assay, which is abbreviated from 5' rapid amplification of cDNA end. So before I explain the result, I will briefly introduce you how does the 5' race work. First, they undergo a reverse transcription, which will generate a cDNA. In this step, they require a primer that will recognize a non-sequence of a target DNA. So the primer is also called a specific primer. Here it is the SP1. And then they treat them with an RASH, which will degrade the target RNA and left the cDNA. And the third step, we require the enzyme called TDT, which will add a string of identical nucleotide at the 3' end. And then we perform a PCR, which require a primer and a second specific primer to amplify the cDNA to further undergo other investigations. So the product of 5' race can provide the sequence of an RNA transcript. Here, in this example, the 5' race product represents the RNA fragment that was cleaved by AGO2. The author first predicted that the AGO2 cleaved RNA at this site. And then they sequenced the 5' race product. As they expected, the product sequence are exactly the same sequence as they predict. You can see the target RNA from this site, UGG, along to the GUG. Because the RNA is converted to a cDNA, so the U is converted to TGG along to GTT is identical. So we can see after siRNA treatment, here it is SI-MALAT1 that generated a 5' race product indicated by the arrowhead. And for comparison, the SI-luciferase treatment or no reverse transcriptase or no treatment will not generate any of the 5' race product. And then to further test the potential for nuclear AGO2 to cleave the RNA substrate, they perform an in vitro cleavage assay. In these assays, the cells are either transfected with SI-luciferase or SI-luciferase mismatches. And then they isolate the fraction from either the nucleus or cytoplasm. And the third step is to immunoprecipitate the AGO2 protein from the nucleus fraction or cytoplasm fractions. And then they will mix the AGO2 protein with a radio-labeled mRNA. Here, it is the radio-labeled luciferase mRNA. So, because the SI-luciferase can specifically target the mRNA, so they generate a cleaved product shown here. While the SI-luciferase mismatches will disrupt the silencing activity, they will not generate any cleaved product. So in the nucleus, It's a similar story that are present in the cytoplasm. The Si luciferase will generate and cleave product shown here, while the mismatches won't. And sorry. So here, the T1RS treatment will cleave the guanosine of the RNA. So they act as a ladder, while the alkaline hydrolysis will cleave every single nucleotide of RNA. So they generate a series of bands. And then, to visualize the RNA-mediated activity in the nucleus, they perform a fluorescence in situ hybridization before or after the siRNA treatment. In the upper panel, we can see that the FISH assay performed before siRNA treatment shows the MALAT1 in the nucleus. Here, the nucleus is stained by the DAPI. While in the lower panel, we can see that the MALAT1 are not present in the nucleus after the siRNA treatment. So I will make a small conclusion here. There are several evidence to support the idea that RNA slicer activities are similar in nucleus and cytoplasma. First, they quantify the RNA that were presented in the nucleus and treat them with siRNA. The result shows that after the siRNA treatment, the nuclear RNA are reduced. And then they perform a 500 assay, which they predict the RNA cleavage site. And as they expected, the RNA are produced as they expected. And the third step, they perform an in vitro cleavage assay, which they isolate the AGO2 protein from the nucleus extract. And the AGO2 protein in the nucleus are performing a similar function in the cytoplasm. And then they visualize the RNA that are present in the nuclei by the FISH assay. So after seeing that the siRNA are present in the nuclei, the author want to evaluate the localization of microRNA in the nuclei. Here, they identify 456 microRNA in the whole cells, and within them, there are 346 microRNA in the nucleus. What's more, the majority of microRNA in the whole cells are the same in the nucleus, indicate that there are similar distribution of microRNA in the cytoplasm and the nucleus. And then, to examine the association of microRNA with AGO2 protein in the nucleus, they immunoprecipitate the AGO2 protein from the nuclear extract. Here, the IgGs act as an active control. And these results reveal that there are numerous microRNA associated with AGO2 protein in the nucleus. So to further investigate the potential for microRNA pathway to operate in the nucleus, they perform an in vitro process assay to test the ability of dicer. Dicer, as we know, is an enzyme that can process precursor microRNA that will cleave their stem loop structure and generate a mature microRNA that was loaded on the RISC complex. So the first step in the in vitro processing assay is to isolate the dicer from the nucleus or the cytoplasm. And then they mix the dicer protein with a radio-labeled precursor microRNA. In this experiment, it is precursor micro 19A. And then they will analyze the cleaving product of the precursor from the precursor microRNA to see which will represent the dicer cleaving activity. So in this example, the dicer collect either from the cytoplasm or the nucleus will generate expected 23 nucleotide product. So these results suggest that the dicer can process precursor microRNA in the nucleus.
In previous examining AGO2 slicer activity in vitro, the extras were initially prepared from cells that were transfected with the siRNA. And this protocol required the RNA to pass through the cytoplasm before entering the nucleus, and therefore loading might occur in the cytoplasm, and the nuclear import the complex to do the slicer activity. To determine where loading was occurring, they added small RNAs directly to nuclear extras prepared from untreated T cells, and subsequently performed in vitro assays for slicer activity. As they have observed previously, when AGO2 is isolated from extract of cells transfected with siLuciferase, AGO2 from both the cytoplasm and the nucleus can catalyze cleavage of the luciferin substrate RNA. By contrast, extracts from untransfected cells which double-stranded siLuciferase was added after the extract preparation revealed that the cytoplasmic AGO2 was able to catalyze the cleavage, but the nucleus AGO2 was inactivated. And these data suggest a deficiency in AGO2 loading in nuclear extracts. They further examine the loading of single-stranded guide RNA alone. The results show that the single-stranded siLuciferase show low but similar level of both the cytoplasmic and the nucleus AGO2. And the RNA single-stranding can be directly bound by AGO2 without the need for additional factor as a control to ensure that the nuclear AGO2 was accessible for loading. They immunoprecipitated AGO2 and then incubated it with a radiolabeled single-strand siLuciferase RNA. They found that both the cytoplasmic and the nucleus AGO2 were able to bind a single-stranded guide RNA, as you can see in the figure B. To more directly investigate AGO2 loading with the duplex small RNA, AGO2 was immunoprecipitated with the antibody against either the endogenous, as you can see in the figure C, and the free-taking AGO2, as you can see in the figure D. They observed the co-purification of the radiolabeled siLuciferase guide strand were cytoplasmic, but not in the nucleus. These data suggest that the AGO2 from the nuclear extracts were first mixing with the single-stranded but not the duplex RNA, and second, loading of the duplex RNA being deficient in the cell nuclei. In small duplex RNA loading involves an initial step when the duplex first binds to the RISC and then begin to unwinding step when the passenger strand is removed. To determine the limiting step, whether it's binding or in unwinding with the nucleus loading, they perform the in vitro AGO2 loading assays under condition. They will discriminate between the duplex siRNA or single-stranded guide RNA bound to AGO2. When RNA isolated from the AGO2, immunoprecipitated was resolved. Both the duplex and the single-stranded RNA were bound to the cytoplasmic AGO2, indicating that the formation of both the site complex during loading. In contrast, neither complex was formed during loading in nucleus extras. And this result demonstrated that the initial state of the duplex RNA loading is deficient in nuclear extras. And then they observed of the microRNA and dicer activity in cell nuclei before. So then do the examine loading of the microRNA. Like fully complementary RNAs, they observed that the mismatch of the duplex were loading in the cytoplasmic extras, but were not loaded in the nucleus extras. But the microRNA can show both loading in the cytoplasmic and the nucleus. So this data show that the microRNA can load in both the cytoplasm and the nucleus, but the siRNA can only work in the cytoplasmic. Because several proteins are implicated in the AGO2 loading and the RISC maturation, they perform the Western blot to detect their presence in isolated nuclei. Surprisingly, these loading factors were all exclusively cytoplasmic. The only exception is HSC70. This protein is functioning in open the AGO2 to that the RNA binding. So this result shows that there should have some critical RNA factor within the nucleus, so for forming the multi-protein complex. And next, move to the conclusions. So at first, the author used microscopy and a new cellular fractionation protocol assure that the AGO2 and the dicer TRBB and the TNR62A are all present in the human cell nuclei. Next, they use co-IP to prove that the RNAi factor can form a stable multi-protein complex in nucleus.
Next, they use the 5' race and the fish, showing that RNA is active in the human cell nucleus. Next, the author showing that the microRNA are bound to the nuclear AGO2 and the microRNA pathway component are in the nucleus. And finally, they find out that the siRNA is deficiencies in nuclear because of the necessary risk loading and maturation factor are absent from the nucleus. So these papers show that previous RNA research are based on the cytoplasm, but now the author find out that RNAi can also present in nucleus. So do RNAi take part in the transcription or splicing mechanism? It can be further explored. And this is our presentation. Thank you for your listening. Do you have any question?
Thank you for your great presentation. I wanted to ask that in this particular paper, it is shown that the risk-loading co-chaperones are absent in the nucleus. So there are, in other words, there are they are specifically localized in the cytoplasm. So what do you think will happen if we create a cell line that will have all these proteins localized also to the nucleus? What would happen to the to the mRNA and siRNA activity if all these factors are also translocated in the nucleus? Thank you.
Well, I think that the siRNA and microRNA that were present in the nucleus may undergo some gene expression without exporting those proteins out of the cytoplasm. So they can directly alter the gene expression, such as the chromatin or some mRNA that are present in the nucleus. So the microRNA and siRNA present in the nucleus have such directly effect in the genome.
Do you think it will be possibly lethal for the organism if they have all these factors also localized in the nucleus, in both nucleus and cytoplasm? Because it might increase the activity of both siRNA and mRNA pathways. So is it possibly lethal for the organism if these silencing activities are happens both in the nucleus and the cytoplasm?
Let me refresh your question. Sorry, previous student probably really catch your question. So the question is if in the last figure showing the all those chaperone proteins are only presented in the cytoplasm, but if they change or let's say we do some recombinant protein that it expressed in the nucleus, what happened to those microprocessor or the microRNA? Can the risk loading can happen in the nucleus as well? And also, is there any lethal effect on the cell if we overexpress those chaperone protein, which may involve in risk loading in the nucleus, just overexpress in the nucleus, what will happen? Is that your
That is
question?
my question.
Is that
Yes.
okay?
I'm sorry.
Answer? No? Yeah, I think this is a good question because nobody really tested if the overexpression in the nucleus can change. I think that it will change the microRNA profile in the nucleus versus in the cytoplasm. And also maybe this is a good way to test if which protein is required for the risk loading in the nucleus, because we can just add one in the nucleus one by one and see which one increase the nuclear risk loading activity. So, like for people who are really interested in this field, maybe can try this in the follow-up experiment. Yeah. Okay, thank you. Is that okay? Sorry. Any other questions? No? I have a question. Could you go back to the slide for figure 6F? Yeah. Do you think the human mirror 19A, the process product in the nucleus is the same as the cytoplasmic one?
I think it's different because the molecular weight is different. The band is different between the cytoplasm and the nucleus. So there might be some different component complex for siRNA being function or the microRNA being function.
Speaker. So, sorry, I forgot there's a speaker on this by the seat. So I was asking that the nuclear processing, the risk loading for the human mir 19A looks like the size is different as compared to the cytoplasmic risk loading result. So do you think this data really support the conclusion that only micro MI risk loading is sufficient in the nucleus, or it could be other mechanism? Or if you are the reviewer, you will ask the author to do some kind of a control experiment. Just give some suggestion. You think this data is strong enough to convince us?
So you can be to check that there's some microRNA is loading, some others microRNA is loading on the cytoplasm or the nucleus, and there will be different too. That the band is, yeah.
Okay, thank you. Because for me, it looks like the nuclear and the cytoplasmic product, the mere 19A is almost like a 4 or 5 nucleotides in difference in the size. So it's kind of interesting, maybe from other, like I said, trimming mechanism or something else. So we don't know. Yeah, thank you.
I have other questions. No.
Is there any question from Kyoto and Tsukuba?
Oh, hello. Actually, this question might be slightly related to the other question that was asked in Kyoto, but
Yeah.
the imaging, the microscopy imaging, the cells are fixed, and so they're not live cells.
Yeah, those
like
are fixed cells. Yeah.
So is there any it's quite similar, but it show the relative quantity or the distribution between in the nucleus and the cytoplasm, but do you know if there's if it's live imaging or time lapse, is there's translocation or any significance? Did they mention anything
You mean the
of these proteins?
this protein, the translocations?
I mean, this is already fixed when they image. Yeah.
Yeah. So yes, those are fixed image, but I don't think they really provide any live image of the translocation shuttling from the cytoplasm to the nucleus. As also in my lab, we try to monitor that, but because maybe you can use the fluorescence tag, the protein, but the fluorescence tag protein is kind of overexpressed. So we don't know it's really represented the real quantity of the protein in the cytoplasm. Is there any overexpression effect? But I have to say, when I read this paper, I think the bottom, like this slides, the bottom two slides, I think is not real. Just too beauty to me. For me to really think of it as a knockdown effect of SI-MALA1. Because actually in our lab, we repeat these experiments. Well, I don't know. It's kind of like a really beauty figures for people to think that is really knock down or that completely knock down or destroy those MALA1 long non-coding RNA. Okay, any other questions?
Okay. Thank you.
Any other questions? Anyone from Taiwan? No? If no, that's I think it's about time. Let's thank these 5 students for today's presentation, and thank you for all the people from the Tsukuba and Kyoto and all the students in here. Thank you.
Thank you very much.
Thank you.
Thank you very much.
Thank you very much.
Thanks.